Hi, my name is Tegan Kuzner, and I'm an upcoming junior studying environmental microbiology. I work in Dr. Emma Morrissey's lab in the Division of Plant and Soil Sciences. Today, I'm going to present on my project focusing on the response to climate warming and Boreal peatlands. So first, wetlands are ecosystems that are flooded by water, either permanently or seasonally, where anaerobic processes prevail. This means that processes that are without oxygen prevail. So peatlands are a terrestrial wetland ecosystem in which the production of organic matter exceeds its decomposition and a net accumulation of peat results. So the year-round waterlogged conditions that cause continuous saturation leads to an anaerobic conditions that slow plant decomposition so much that there is a large accumulation of plants. And this large accumulation of plants is peat. So peat grows vertically around 1 millimeter per year. So that means that 1 meter of peat needs 1,000 years to form. The focus of this project more specifically is bogs, which are raised above the surrounding landscape and receive water only from atmospheric sources and are not in contact with groundwater. This means that they are often nutrient poor and very acidic. Peatlands are usually associated with fresh water and cool or wet climates, and they serve many different ecosystem services. First and foremost, peatlands are carbon sinks, meaning that they contain large amounts of carbon. So peatlands have sequestered 200 billion tons of carbon worldwide and contain 42% of all soil carbon in the world. Bogs also absorb precipitation and runoff so they can help prevent flooding and droughts in certain areas. They also perform many recreational and educational purposes, such as in Cranberry Glades, West Virginia, which is actually the focus of this project. So by 2050, climate warming is predicted to cause increased greenhouse gas emissions as a result of microbial metabolism. So this increase in decomposition rates can lead to the release of CO2 and CH4, or carbon dioxide and methane, which would cause the bog to transition from a carbon sink to a carbon source. So for vegetation, sphagnum mosses and vascular plants, which are native to wetlands, have chemically distinct properties and root activities that may potentially influence microbial community composition and function. So warming is likely to shift the vegetation from sphagnum moss to vascular plants. This is especially important because these plants, again, have distinct properties. For example, sphagnum moss can acidify its surroundings by taking up cations such as calcium and magnesium and releasing ions. Sphagnum moss can accumulate nutrients in its tissues to reduce the availability of nutrients for other species. So this transition from sphagnum moss to vascular plants is predicted to alter carbon biogeochemistry and microbial processes. So that brings me to the focus of this presentation, this project. So we did a warming experiment in Cranberry Glades, West Virginia. So we sought to determine how temperature affects aerobic and anaerobic microbial biodiversity and activity in Boreal peatland soils. We also sought to determine how specific wetland microbial species have variable growth rates and different temperatures. And then finally, we sought to answer if vegetation type influences carbon mineralization rates under different warming conditions. So as I mentioned, this experiment was conducted in Cranberry Glades, which is the southernmost boreal bog in North America. So this means that it has species that are found in northern bogs, such as in Canada. So in terms of vegetation, they do have cranberries, which is what they're named after, sphagnum moss, as I mentioned, skunk cabbage, purple pitcher plants, and sundew. So it is at a relatively high elevation, so the ecosystem is fairly different than the rest of West Virginia. And the pH is around 3, which, as I mentioned, bogs are fairly acidic because they only get precipitation or water from precipitation. So our experimental design. So we had 4 separate plots, as you can see here, and we sought to have a vascular plant and moss in each of the plots, and we sampled around those. So we had 3 different treatments, temperature treatments, so 15 degrees, 25 degrees, and 35 degrees Celsius. And we started out with a soil incubation experiment. So 15 degrees represents the average summer low temperature in Cranberry Glades, while 25 degrees represents the average summer high temperature. The 35-degree treatment represents the effects of global warming. So we have completed a dual incubation for carbon mineralization and H2O water, which is heavy water, and we're completing quantitative stable isotope probing with this. And we have done that aerobically and anaerobically among the different temperature gradients, so the 15, 25, and 35 degrees. So here are results. So this shows the carbon dioxide respiration measurements, and as expected, they all increased. You can see here that the higher temperature treatments, so the 35-degree, had higher respiration than the 25-degree, which was higher than the 15 degrees. Interestingly, the anaerobic was a little higher than the aerobic, which is a little abnormal because the anaerobic species or the anaerobic microbes typically take longer to grow. So here we have the methane respiration measurements, and this is just for the anaerobic. And as you can see, it had a similar trend in that the higher temperature had the higher cumulative methane. So here are our biomass measurements. As you can see, the anaerobic samples had significantly higher biomass measurements than the aerobic. So this means that they were storing more carbon than the aerobic. And as you can see, this also did, at least in the anaerobic treatment groups, increase with temperature. So we also looked at ammonia, nitrate, and nitrite. So here is the ammonia. And as you can see, it did increase with temperature gradient. And interestingly, the anaerobic did have higher ammonia concentrations or average concentrations than the aerobic. And here are the nitrate and nitrite. So there's nitrite on the left, or A, and then nitrate on the right, or B. And these show a little bit different things than the ammonia, so in terms of the relationships. So for the nitrite, the anaerobic seems to be higher, except for maybe in the 15-degree treatment. However, with the nitrate, there's kind of an inverse relationship between the nitrate and treatment in terms of the aerobic and anaerobic. So in the aerobic treatment, the average nitrate decreases with temperature. However, in the anaerobic, it increases with temperature. So to sum that up, we found that temperature increases are associated with increases in both aerobic and anaerobic respiration. We also found that anaerobic samples seem to have higher biomass measurements than aerobic samples. And then finally, we also found that aerobic and anaerobic samples may utilize nitrate differently as they have inverse relationships with nitrate and temperature. So this project is still ongoing and we still have a lot of data left to collect. However, the data that we've already collected and the data that we will collect will help to create a better understanding of how microbial communities respond to warming, especially in anaerobic environments, which is less studied. And so this is essential when determining how these environments will respond to global climate warming. So mitigating these microbial responses is vital to counteract the impacts of climate change, such as warming, more extreme weather patterns, and rising sea levels. So that concludes my presentation, but if you guys have any questions, please do not hesitate to reach out. Thank you so much for listening.